Assalamu alaikum everyone. My name is Ahmed Uzair and I am part of the Faana board. Faana stands for Fast Alumni Association of North America, and we are here to conduct our fifth session of our recently started webinar series. And today's topic is medical imaging and its practical applications. Our distinguished guest speaker today is Dr. Arish Kazi. Before delving down more into his work and his background, I would quickly like to go over the information about what Faana is and what do we do. Faana basically was started almost 5 to 7 years ago with the main aim to raise funds for different initiatives on FAST campuses all across Pakistan. But so far, our initiatives have focused towards two main things, which has been scholarship fund and research fund. Under scholarship fund, we have been supporting needy students via the campuses and raising funds and collecting funds which would eventually go to the campus, and then campus would pick the deserving students and help them complete their studies. The second initiative has been on the research side, where Faana has been helping the campuses take initiatives that encourage students to go towards the R&D. And in this, we have also been helping various campuses set up R&D labs and incubation centers. So far for 2019, our main focus has been digital engagement via the webinars, which this particular event is part of. Then we have also been involving enhancing North America alumni membership. We've been doing a lot of campus outreach activities. We've been involved in updating and streamlining Faana digital channels. We have WhatsApp, we have Facebook groups, we have LinkedIn groups, so all those various channels. And then the financial planning, which is obviously a big part when you're raising funds for various campus activities. This is a quick snippet of our financials. We have allocated $52,000 for scholarships at 5 campuses and 5 software competitions. For scholarship funding, so for scholarship funding, we have $1,200 per student per semester if somebody wants to donate, and then if somebody wants to sponsor a student for an entire year, it is $2,400. For the research fund, a software competition can be sponsored for $1,200 for an entire. Looks like so. Sorry for the interruption, but so I was saying about so the software competition can be sponsored for $1,200 per event. Lastly, these are our various digital channels that I was mentioning before. Uh, we have uh, we have Fast NU Alumni Association, the official website faana.org. We have a WhatsApp group. You can join that WhatsApp group by texting the message to this number, which is +1 248 697 6194. We have a YouTube channel where this webinar will be posted afterwards. Uh, we have a LinkedIn group. We have North NU alumni emailing list, and then we have Facebook groups and pages as well. Which, if you have any sorts of question, you can reach out to me later on, and I would be more than happy to talk to you more about them and how to get more engaged. Now, going back to our current session, I would briefly go over what this session is going to be about. So, our medical imaging has lately revolutionized the clinical decision making. Magnetic resonance imaging, computerized tomography, and other imaging modalities have enabled clinicians to non-invasively and reliably visualize internal organs and provide important information about the psychological and pathophysiological physiological changes. To talk more about this, we have our distinguished speaker today, Dr. Arish Kazi, who has more than 15 years of industry experience performing medical imaging analysis and processing research and development, both at private companies as well as in hospital settings. So with that, I would like to pass over the ball to Dr. Kazi. And I think, yeah, here you go, Dr. Kazi.
Okay. Thank you, Zaid. Can you hear me?
Yes, I can hear you, and I can see your screen as well.
Okay. So hello, everyone. I welcome, my name is Arish Kazi, and I welcome you all to this talk. So a bit about me. I have a PhD in computer science, where the majority of my research work during my PhD was in the field of medical image analysis. I'm currently working as head of R&D at Claronav, which is based in Toronto, Canada. Claronav is a company that makes medical devices primarily for image-guided surgery. And in the past, I've worked in different companies, most prominently at Philips Medical Systems. I've been working in this industry for about 15 years. I wrote my first commercially used medical image segmentation algorithm in 2004. In this presentation, I'll primarily focus on how the field of medical image analysis is impacting healthcare by using examples both from my past and my current work. But before we move on, let's look at some background. So imaging has become an essential component of medical research. So the goal of medical imaging is to facilitate clinical decision making. So let's see how it's done. So this is an image, an MR scan of the brain, which shows a tumor over here. So as you can see in this image, that without cutting up, it enables the clinician to non-invasively see the underlying anatomy. And the market of medical imaging is going to shoot up to 49 billion by 2020. And with such a huge market, there is a great demand, obviously. With the advent of all these instrumentation, it has led to an immense amount of data generation. So imagine a clinician manually segmenting this tumor for 100 patients. It'll take ages. And the thing is that the amount spent on contouring or delineating information about the tumor can very well be spent on seeing other patients. Now, this is where computer science and automation comes in. So we as computer scientists in this field, we develop automated methods that can both qualitatively and quantitatively analyze these medical images. Hence, this field is medical image processing and analysis. So let's look at, what kind of different imaging techniques that we have available in this field. So it all started with the discovery of X-rays. So this guy, Willem Röntgen, did his first X-ray scan in 1885, and this is a scan of his wife's hand. This is a typical 21st century X-ray image. Now, there are other modalities such as computed tomography or CT. Now, CT uses the principles of X-rays. However, these X-ray measurements are taken from different angles, which produces cross-sectional or 2D slices of the scanned object, and then these slices are then processed further to generate a 3D volume. So X-ray, regular X-ray results in 2D images, whereas computed tomography images are inherently 3D images. Then we have MRI, or magnetic resonance imaging, which is again a 3D imaging technique. And MRI, in contrast to CT, is based on the principles of nuclear magnetic resonance. So what it does is that it measures spatial variations in the phase and frequency of RF pulses or radio frequency pulses that is emitted and absorbed by an object in the presence of an external or uniform magnetic field. This is a typical MR scanner. So this board over here houses the external magnet, and this magnet is actually always on. Then we have ultrasound, which actually uses sound waves, and it's a very common imaging technique prominently for monitoring fetal growth. Then we have PET and SPECT. PET is positron emission tomography, and SPECT is single photon emission computed tomography. They're both based on the CT technique. However, they rely on a radioactive tracer being injected in the human body. So let's look at some of the images. This is an X-ray image of the thorax region. This is a CT image of the thorax, and these are MR images of the brain. Now, a question that you may ask is, why so many different imaging techniques? The thing is that they provide different kinds of information. So X-ray and CT is actually good for showing bony structures. Whereas MRI is really good for showing soft tissue contrast, such as that in the brain. Now, another important thing here is that CT is based on X-rays, and that radiation, X-ray radiation, is ionizing radiation, which means that it's harmful for the human body. Whereas MR is non-ionizing radiation, which means it's very safe as long as you don't have metal inside the body. Now let's look at what the field of medical image processing and analysis, can attempt to answer some of the questions. Can we improve the images? So when these images are taken, they may contain noise. Now the noise may be due to the underlying instrumentation being used, or maybe other factors such as patient motion. So one of the things that we can do is using these algorithms is to denoise those images. So the idea is to reconstruct the original image from its noisy observation, and while we want to also preserve important details about anatomy. So we want to denoise the image and such that it only removes the noise, and we don't want to distort the underlying anatomy. Then another thing that we can answer is, can we extract essential information? So this is also called as segmentation. So segmentation, basically what these algorithms do is they divide the image in regions to highlight important areas, such as, where is the tumor, or it automatically goes and highlights the region in the image where the tumor is present. Can we assist in diagnosis, also called as computer-aided diagnosis, such as divide the image and highlight regions which are cancerous? Can we obtain quantitative measures? Can we develop methods that can tell me, okay, what's the size or the volume of the tumor? Can we combine information from multiple data sources? So, for example, I talked about MRI and CT, and these both images provide different information. So is there a way, can we fuse the data from these both images? And this is done through image registration. So what registration does is that it takes it creates a transform that maps locations from one image to corresponding locations to the other images. It's kind of takes these both different two coordinate systems and maps it into one coordinate system. Now, an example over here, this is a smart 3D denoiser that was developed in our company. Here is an example of an image with noise, and you can see that after the noise removal, we see something like this. So this algorithm is completely automatic, and it leads to approximately 600% increase in signal to noise ratio. Another example of the denoiser, here you can see that the image is completely useless. You know, you cannot really see the underlying anatomy over here, whereas after the denoising, at least this can be used for further diagnosis. Another example for CTA bone removal. So CTA is computed tomography angiography. So it's actually a CT technique that's used to visualize arteries throughout the body. So what happens is that a contrast agent is injected into the blood vessels, and the idea is to look for blockages. Now, in these scans, these arteries are obscured by the bone. So the idea is to develop a method which can actually remove the bone such that these arteries can be visualized. And this technique was developed where you can see the result over here. So the remaining part of my talk, I will actually look at some of the methods, specifically how diffusion imaging is used for mapping the human brain, how MRI can be used for understanding the progression of osteoarthritis, which is actually a joint disease. We look at how computation is helpful for radiation therapy planning. And we look at some applications in Hello?
Yes, we can hear you now.
Oh, sorry. When did my voice get disrupted?
Yeah, towards the end of this slide.
Okay. Yeah, this is the outline of the presentation. So we're gonna go in. Let's start with diffusion imaging. So at the heart of the brain nervous system are neurons. So neurons are nerve cells that transmit electrochemical signals. So this is what a neuron looks like. So it's divided in two parts, the cell body and the axon. So the cell body, it actually integrates information from other neurons, whereas the axon, it actually transmits the information. Now, the axon is surrounded by a fatty substance called myelin. Now, myelin is white in color, while the tissue containing the cell body is colored gray. Hence, dividing the brain in two main parts, the white matter and the gray matter. And you may have heard these terms before. Now, these are some of the white matter tracks, most prominent white matter tracks in the brain. We are mostly, in this talk, we look at the corticospinal tract, which is actually the largest descending pathway in the human brain, and it's responsible for providing motor functions such as limb movements. So these tracks go through the spinal cord and they connect to the motor cortex of the brain. Any damage to these tracks can actually lead to paralysis. Now, how does diffusion? How can we use diffusion imaging for this kind of visualization? But before we look into diffusion imaging, what is diffusion? So imagine that you have a glass of water lying on a table in a sealed room. So that water may seem stationary to you, but at the molecular level, the water molecules are moving. Their motion is random, and this is also called the Brownian motion. And this is what diffusion imaging aims to quantify. An important property about diffusion in white matter of the brain is that it's anisotropic, which means it's direction dependent. So there is faster diffusion along the axons than across it. Now, Peter Basser in '94 developed a mathematical model. So what he did was that at each location of the image, of the diffusion image, he quantified a tensor. A tensor is a mathematical entity. It's in layman terms, it's like a 3x3 matrix. So he quantified a tensor at each location, and then the tensor is then diagonalized, resulting in three eigenvectors. So the idea is to use the tensor to map these pathways in the brain. So a single trajectory through the brain is generated by connecting the principal eigenvector of each tensor. So this technique is called tractography. And so what you're essentially doing is that you can imagine that your principal eigenvector is like a vector field, and you trace the path of a particle in that field, and the tract, the generated tract over here, is a curved tangent to that field. So here's an example of tractography being done in the brain. So this over here, you can see the white matter fiber pathways of corpus callosum. Now this is the largest white matter fiber bundle in the brain. And that example, this was work with L.J. Donnell at Harvard Medical School, and you can see all the this is a result of full brain tractography. Now, why is this information important, or what can be it used for? So this is the image I showed earlier. So these are the corticospinal tracts, and you can see that they are connecting to different areas of the motor cortex. And this is how, with the current technique, how the spinal tracts were visualized based on tractography. Now these blobs over here, what do they represent? These are actually fMRI activations. So what happens is that the patient is given a task such that, move your hand or leg or face, the limbs, and then the fMRI instrumentation, it goes and it measures the hemodynamic response or changes in blood flow, and then it maps the areas of the brain which are active during that task. So you can then visualize, okay, which areas of the brain were active. So these this area of the motor cortex actually reserved for the hand. This area is reserved for the leg. So the whole idea is, with the standard tractography, we can visualize tracks going to these areas. And I'll, in a minute, show you why is this important. And as you can see, that we know that the tracks should actually go and diverge over here. So essentially in this area, there are several tracks and they are crossing each other. Whereas with the standard technique that was developed early on, it's just not possible. Now, what we did was we developed a technique that can actually go ahead and resolve fiber crossings. So this is the standard. This is an image showing the standard technique, and this was a technique that was developed. And here you can see it can resolve all these tracks over here. And again, why is this useful? So for example, a neurosurgeon is resecting a tumor in this region. Now, they want to know which pathways are present over here. They don't want to go and cut the wrong pathway, because this could actually lead to paralysis. Another example over here where with the standard technique, you can see the pathways around the tumor. With the proposed method, which was developed, you can see many more pathways over here. So if anyone is interested, some further info. So this method was actually developed as part of the open source toolkit team, which is part of 3D Slicer. It's a software platform developed by Harvard Medical School, and it's a joint collaboration between the surgical planning laboratory at Harvard Medical School and the MIT CSAIL department. So this is actually a good resource for students who want to pursue, further information regarding this field. It's a very good tool. So now let's look at some methods for how MRI is useful for analysis of the articular cartilage. Now, osteoarthritis is a very common joint disease. It's degenerative in nature. It affects the lives of more than 200 million people with an estimated burden of about $65 billion. So the prevalence of OA increases with age, and currently there is no known cure for OA. The current treatments, they're directed more towards pain relief, and OA is generally characterized by degeneration of the cartilage. So what's the cartilage? So this is your lower bone of the knee, which is the tibia bone, and this is the femur. Now, cartilage is a region in between these two bones. So this is an image of what bovine cartilage looks like. So as you can see that it's a frictionless gliding surface, and this is what gives the joint its mobility. Now, what happens to the cartilage during OA? Let's look at that. So this is a healthy joint, and this is a tissue of the cartilage seen under the microscope. Now, as the disease progresses, you can see that the cartilage starts to break down. And during the late stages of the disease, the cartilage is completely gone. And then it's just bone against bone. And this is what leads to pain. Now, the current gold standard for monitoring the progression of osteoarthritis is actually from radiographs or from X-rays. So what radiologists do is that they go ahead and they try to measure the space between the femur and the tibia. And based on the spacing information, they go and rate it according to the scale. Now, and they call this joint space width. Now, is joint space width an optimal measure? Certainly not, because the cartilage is not even visible in X-rays. Now, MRI has been gaining a lot of attention in the assessment of the cartilage. Why? It's because MRI is non-invasive and it allows visualization of the cartilage. So as you can see over here, this is a knee MR scanner, and you can see this is an MR scan of the knee, and you can see that the cartilage is visible over here. And you can develop methods where you can go and create algorithms that can go and automatically segment the cartilage, like the one that's shown over here. Now, based on this, based on the segmentation of the cartilage, you can generate computative methods like cartilage volume and thickness, which can be then used to monitor the progression of osteoarthritis. Now, another image over here showing the segmentation of the cartilage. And this was a framework that was developed to monitor and find out which areas of the cartilage are most affected by the disease. And it was statistical. So what it did was that it went and it took a set of data, a set of knees of healthy and healthy knees and patients with osteoarthritis, and it tried to find out which regions in the cartilage are most discriminative of the disease. Where does most pathological changes happen? And we found out that most of the disease modifying regions are most towards the exterior of the cartilage. Now, why is this information useful? This information can be useful for monitoring the disease progression, or most importantly, it can be extremely useful for clinical trials. So for example, if you want to develop a drug, you want to monitor progression of the disease or the effects of the drug only in regions where it make effect. So you filter out the noisy regions and only look at this region. And indeed, this was what the method was used for. So it was part of a very large clinical trial done by a pharmaceutical company. So anyone who is more interested in learning more about the method, these are some of the resources. Now, let's look at some of the methods that were developed for radiation therapy planning. Now, what is radiation therapy? So radiation is a technique which goes and tries to kill cancer cells. Now, radiation itself doesn't care about healthy cells or cancer cells. It's going to go and kill anything in its way. Now, this is not good because when you are giving radiation to a patient, you want to have minimal damage to healthy tissues. Now, with recent advances in radiation therapy, specifically IMRT, which is intensity modulated radiation therapy, you can actually give a precise dose delivery to the tumor with minimal damage to surrounding tissue. So what you intend to do with IMRT is that you give a very high dose of radiation to the tumor, and then the radiation to the surrounding tissues is not that high. But this is only possible if you can actually go ahead and contour all the structures in that particular case. So you take a CT scan of the region where you want to give the radiation, and then in order to use IMRT, you go and you have to contour all the structures, both healthy and the ones where you have to where the tumor is. Now this takes a lot of time. So as an example, an average physician's working time on a single head and neck IMRT case is like 3 hours. So for 3 hours he spends for one case, and imagine that if that whole method can be automated, then those 3 hours could be spent seeing new patients. And this is what the whole idea was. It was to develop a fully automated methodology for the head and neck region. So these are some of the results for the method. So you can see that these contours over here, this is a head and neck CT case, and it's showing you the contours that was done by the physician. These are shown in green, where the contours that were automatically delineated by our method are shown in colored. Now, these are the different regions. So this is the mandible. This is also your lower jaw. These are parotid glands. This is the brainstem, and these are neck nodes. Now, this method was actually developed as part of Philips' platform called Pinnacle. It's a system for giving radiation therapy, for doing radiation therapy planning, and the method was named SPICE. SPICE stands for Smart Probabilistic Image Contouring Engine. And it has been now in routine used, and the feedback that I've got is really good. Like it's within a matter. Imagine that now that spending 3 hours, you don't have to spend 3 hours. In a matter of minutes, your contours are generated. This is another example of the methodology, and here you can see that all these organs were segmented within a method of minutes. So for some more information, if anyone is interested. So now the remaining part of my presentation will focus on image analysis primarily for dental applications. So I've been developing methods in the dental world now for about 10 years. So an example is 3Shape's Trios. So 3Shape is a company based in Denmark, and we started development of an intraoral scanner. So an intraoral scanner, what the idea was that to enable digital dentistry workflow. So what happens is that when a patient walks in, a dentist actually takes an impression of the teeth to see, if the teeth are aligned, or what. Let's suppose if the teeth are missing, then they take an impression. Now that impression they have to go and then scan, and it's all time consuming and leads to more chair time for the patient. Now the idea was to develop a scanner where you could actually go and directly scan the patient's teeth without taking any impressions. So you go ahead and directly scan the teeth and the surrounding soft tissue. This is what was developed. I worked only on the first prototype of the system, and this is what the scanner looks like. So you see the dentist actually goes and it scans and it generates a 3D model of the teeth and the surrounding tissue. So here's a video just showing you the latest version of the scanner. It's a wireless, and it's extremely fast. So you can see here the person is actually scanning his the lower jaw, and in a matter of seconds, it goes and it scans, takes all the images, stitches them, and you see under 11 seconds the whole lower jaw was scanned. So this is what the power of computing is in this field. So for the past almost 7 to 8 years, I've been involved in the field of image-guided surgery. So it's what is image-guided surgery? It's a surgical procedure which in real time tracks and correlates the position of the surgical tool to preoperative imaging data. Now, this surgical procedure can be human-driven or could be fully autonomous, such as the famous Da Vinci robot for laparoscopic surgery. So what do you require for such a procedure? So you require a set of imaging data, example, MRI or CT data. Then once you have obtained your data, you do a preoperative planning of the surgery. Like where do you want to make the incision? Or where is the tumor present? You know. Now, imagine you have these two worlds. You have an imaging data, which I would call a virtual patient, and then you have a real world which is the surgical world, or the real world where the patient is lying on the surgical table. Now you want to map from the virtual patient to the real world, and this is done through registration. There are different techniques, but I'll talk about the technique that was developed for our product. And finally, once you have the registration, you can go ahead and do navigation. Now, navigation would mean that you try to track the physician's tools and display them in real time onto your imaging data. Now, the system that I'm going to show is actually used for dental surgery, primarily for placing implants. So what are implants? For those of you who don't know, it's a known way to replace missing roots and support artificial teeth. So this is what an implant looks like. So it's made of metal. Titanium is the most commonly used metal, and titanium fuses really well with the bone. And once the implant is placed, then an artificial tooth called a crown can be mounted on top of it. Presently, 90% of the implants are placed freehand, which means the dentist just goes and blindly drills into the bone. Now, this obviously could lead to patient discomfort and risk of infection. Other than that, you have to go and cut the gums to expose the jaw bone. You could, since you are drilling blindly, you could potentially damage nearby nerves or the membranes of the maxillary sinus in the upper jaw. And since you have to cut the gums, you have to go and stitch them up, which extends the surgery healing time. And finally, obviously, since you're doing it manually, it leads to human error, such that the one shown over here. So these were 4 implants that were placed in a patient, 2 without Navigant, and 2 with Navigant. So you can see that the ones without Navigant are actually extruding outside the bone. And this actually is not good because it leads to much less aesthetically pleasing restorations. Now, what is Navigant? In layman terms, it's a GPS for dentists. So using the 3-dimensional cone beam CT as a map, Navigant enables real-time visualization of the dental's drill. It provides guidance for position, angulation, and depth of the surgeon's drill. Our system is very accurate. It leads to the system overall end-to-end system accuracy is 500 microns. So we started development of the system in 2011 as a 2-person team. And the system was co-developed with our clinical partner, which is a UFT dentistry school. Since it's a medical device, you cannot just develop it and start selling it. You have to go and do clinical trials and get regulatory approvals. So the device was approved for sale in Europe in 2015, and then it was approved by FDA in 2016 for sale in the US. So when we started the system in 2011, there was no other system like this in the market. early on in early 2000s, a French company, developed a similar system. However, it was really very expensive and not accurate. So it died out. So now there are a few other systems in the market now, but we are still the global market leader with more than 50% market share. So let's look at the workflow of the system. So the whole thing begins with the patient walking in, and the first step in the process is that you create a stent. A stent is a piece of thermoplastic which get molded around the patient's teeth. Now, with that stent molded, you go ahead and you send the patient to get a scan done. That scan is imported in the software, and the dentist goes and plans where he wants to place the implant. And finally, he goes ahead in the surgery room and places them. Now, this is what the workflow shows like. So you start with the scan, you go to plan, which is your virtual patient, your image, and then you using this mapping or registration, you go to the real patient. So this is where the registration comes in. So to register our system, we use something called a fiducial. So we developed this. It's a piece of metal, aluminum, and it has all these features. And we spent a lot of time designing and testing this. So even in the scanner, so even if you have you know, 3 corners of this fiducial visible in the scan, still you would be able to successfully and accurately register the data. So this is what typical surgeon rooms look like. So this is our system. You have a optical, a stereoscopic camera over here, which goes and it tracks these tools. This is the software that's running. This is our Navigant card. Now, all development of the system was done in-house. So we own the camera, the cart, obviously the software. The software, we are not using any third-party tools. Everything is developed in-house. So this is a small video which actually shows you how the surgery, what happens during the Navigant surgery. So here, a patient, the bridge is removed. The first step is actually going and fabricating the stent. So you put it in warm water and mold it around the patient's teeth. You attach the fiducial to the teeth, and then you send him for a CT scan. So the patient is then scanned, and the data is imported in our software, and then the dentist can go ahead and plan the implants. Now, during the surgery, the first step is to calibrate our tools. This is done in real time and is all gesture-based. So after the tools are calibrated, you know, the dentist can go and just start drilling. And while it's drilling, in real time, you're gonna see the patient's the drill onto the patient's teeth. So you can see that while you're drilling, you're looking at the angulation and the depth and the positional. And here the implants are placed. Now, this system was launched in the U.S. market in 2016. So not you know, not a long time ago, about 3 years. So we started to see some issues. Like the major complaint that we got was the stent. So the molding, although in the video it's not very apparent, but it actually adds 15 to 30 minutes to the procedure time. And the thing is that many dentists, they don't have a CT scanner on site, so they have to send the patient to a hospital. And making the stent is not easy. The dentists, they don't want to do it, and when the assistants do it, they actually mess things up. So for example, this is a stent molded onto the patient's teeth, and you can see the stent is not fitting well, like it's in the air, and this would lead to accuracy issues during drilling. So we immediately reacted, and instead we completely developed a new methodology for registration. So this methodology is completely new. Nobody else has a similar technique or technology in the world right now. So what we did was that instead of using the stent, we said, okay, why can't we use the patient as the fiducial? So we call this technology trace in place. So this happens in real time. And what the dentist does is that he takes this tool and goes and traces around the patient's teeth. So it's it takes under a minute or so to trace the patient's teeth, and then this trace in real time gets registered to the patient's data. So these are some of the examples of the traces being registered to the teeth. We launched this about 1.5 years ago, and so far we have seen quite a bit of increase in the sales of our system. Dentists love it. They don't have to go and get a special scan done. They don't have to make the stent. It's fairly easy, and even if it gets messed up, for example, you go and the registration is not accurate, you just go ahead and do it. So you don't have to go and do any rescans. So to summarize this talk, imaging has become a routine research and clinical tool. Specifically, computational methods such as those for image analysis have become a key component of clinical decision making. So where's the future heading? I see a lot of work in this field, research, as far as research is concerned, in artificial intelligence, AI, or more specifically, deep learning. Now, there's a lot of work going on, but is it being directed more towards commercial products? That's no. And why is the reason? The reason is that these methods, they require tons and tons of data. Deep learning, like specifically convolutional neural networks, they require a lot of data. Now, most of the algorithms that are being researched, they are supervised in nature, which means that with all this data that is available, the physician needs to go and contour these data sets. This requires a lot of time, and this is the time that they don't have. So, and which actually ends up that these methods, they don't have a lot of data. Now, another problem is regulatory limitations. So any algorithm that you develop actually needs approval. You can't just develop a method and go and release it. It needs to be accepted by FDA or European authorities. Now, FDA has been very conservative about approving deep learning systems. And even if they do, they accept the systems to be in the locked state, which means that those systems cannot go and adapt themselves to new data. And if they have to retrain their methods to new data, they have to go ahead and apply for another approval. Will these things change over time? Time will tell. So now we looked at obviously at the field of image-guided surgery and how it's revolutionizing surgery. And I guess industry is moving towards fully autonomous systems such as robotic surgery. And this is something we are also looking at in our company. And that's it. Thank you so much for the talk. If you have any questions, comments, feel free to drop me an email. Thank you so much.
Thank you very much, Dr. Kazi. Really appreciate it. And it was definitely a very informative session. I would be shortly opening the floor for questions to all the participants. But before I do that, I have a quick question. Since I was also listening to the entire session, and it was, a very insightful session for me as well. But so obviously you're a PhD doctor, and it's a very specialized field. So can you talk a little bit about how you started at FAST? Probably you were there for a bachelor's, and how was your career progression from as a FAST student doing a bachelor's, and over all these years building up to this very specialized field. So if you can, for the existing and current students, if you can talk a little bit about that career path or the track that you took that eventually led you to over here, because this is not a very not a common field. It's a very specialized one.
Right. Yeah. So during my degree in FAST, so I'm 99 batch, graduating in 2002.
FAST Islamabad.
FAST Islamabad, right. So my final year project was actually related to image processing, and it was obviously 2D image processing, but that's where it all began. So my final year project was related to using iris recognition for biometric authentication. and that's where my interest in image processing started. And but medical imaging came a bit later. So when I moved on for my master's, um, that's where I actually got an opportunity to work for Philips Medical Systems in Germany, and as an engineer. And there, that's where, you know, since I had a background already in image processing, I actually could go and I was able to get that position, and that's where it all started. Now, as far as the undergrad or even at the graduate level, these courses are concerned in terms of you know, the field of medical imaging, I do understand that in Pakistan it's not very common. And you see, the reason is because, first of all, industry is not very active over there, and secondly, what is the reason, what drives all these methodologies is the healthcare. Now, in hospitals in Pakistan, obviously you have a lot of data, but the physicians over there are not really interested to you know, provide the data to the institutions, or even help them doing this research. So that's why things are really lacking over there. But um, yeah, my primary, like, obviously all this didn't really start back in Pakistan, but my, to answer your question, my basic the reason why I went into this field is because of my interest to image processing and my
And
FIP
your final year project, correct?
and my final year
Okay.
project, right?
Okay, that's good. We just got a question from one of the participants, and the question is, how can your organization collaborate with various professors in Pakistan that are in the field of image processing? Is there a scope for joint venture with the organizations in Pakistan?
Yeah, that's, my organization is actually a private company, and we are not really you know, actively collaborating outside the company. So from that respect, there is no such you know, even our collaboration here is very limited. So to answer your question, you know, it's, but I am available. Like, I some one of my colleagues, he actually now is a professor in an institution in Islamabad, and you know, routinely, off and on, I do get to let's say collaborate with some of his students who have an interest in image processing and medical imaging. So last year I was an external supervisor for a PhD dissertation. So in that way, I'm doing it on my you know, on an individual basis. But from my company's perspective, you know, they're
Okay.
not interested. So
Okay. And then another question from my side is, how easy or difficult was it to, learn all the medical jargons? Because uh, you were and you are still a computer scientist or an engineer, but from your presentation, it looks like you're half medical doctor as well now, or a
yeah.
dentist. So how easy or difficult was to, learn all those stuff?
Honestly, it wasn't very difficult. Like, I just read books, and like, you have to do a lot of reading. Obviously, I mean, there's no shortcut there. So you have to read books. You have to, you know, understand the anatomy and understand from, you know, look at a lot of images, ask a lot of questions. So that's what I did, I guess.
Okay, good. We have some more questions. We have Mr. Ahmed Malik online. Mr. Malik, would you like to ask any question?
Um, Assalamu alaikum. You asked the questions that I had in mind about his journey, and I also heard a great deal about how this business knowledge is important to learn uh, along with the field of computer science to help. So you've asked both of my questions there. Um, any advice you would have for us or the students to work with perhaps medical students in Pakistan to tell them how their work could help eventually the the universities there? I mean, I think, is there an opportunity for medical students and computer sciences students to collaborate to do some joint final year projects that would be beneficial for both the industries?
Yes, definitely. I mean, there is a lot of potential. As you can see, how automation can actually reduce the burden, and especially in Pakistan, where the number of patients significantly outnumber the number of physicians available. So I think there is a lot of potential over there. Now, obviously, this needs to happen both at the institutional level and at the other institution, which is your hospital. I guess it should start at the professor. Whoever is interested or in this, then they should go and they can present a case study to the hospitals and see if they are interested. But you see, from what my experience have been initially is that the level of motivation in terms of the institution is high, but the level of motivation in terms of the institution, the healthcare institution involved, is not that high. And it's not I guess it's primarily because of the burden. There are so many patients, and they don't have time to go ahead and do contouring. It requires, doing this requires a lot of involvement from the clinician. So it's a tough thing to do. But I guess if somebody presents a very good case about, they can from, so even if the clinicians involvement is not that much, still I guess at the educational institution level, the professor himself can be more motivated and maybe help the clinician. So I guess in that respect, the idea is to, you could go and for example, the professor can go to the institution, to hospital, and see where most of the imaging is being done, and then see what kind of problems they have, and start small. They don't have to go and develop a whole product. So they can start small. They can help them with delineation, or help them with let's say if they're looking at radiographs, so they can make a very small software and which can actually go and just mark some locations on the image. So I guess they could be done a lot, but it really, you need involvement from both sides. So I hope that
Yeah,
answers.
I do think, thanks for the answer. I think there is an opportunity between the students at the computer science institutes like FAST or others to work with the students in the medical field to start changing the mindset, so that eventually when those other students go into the industry, they bring in the progressive thought process and progressive mindset, so that eventually those students becoming doctors would start to collaborate with the universities, the students, and other computer science institutes to start that industry and academic partnership.
Right.
Uh, I have another question from one of the
So
participants.
thank you for answering my question.
Welcome, Ahmed bhai.
I have one more question from one of the other participants, Mr. Sayyed Shahriyar. He has actually asked two questions. First one is, how much is it necessary to have a medical background to work within this field? And then the second question is, for a beginner to start working in this field, what are the factors they need to focus on? So I think probably at the bachelor's level. So what factors they need to focus on?
Right. Yeah, you don't really need any medical background. Like I didn't even take biology during my O levels, so you really don't need any medical background. So when you get into this, you start reading books, and it'll all come to you. Now, as far as undergrad is concerned, and this field, your math needs to be really strong. So courses like linear algebra or numerical methods, you need to be really good at those, because those become the foundation of what I'm doing right now.
Okay. Okay. Is there anybody else with a question that he would like to ask? You all guys are unmuted right now, so you can ask a question live as well. Okay, so looks like I think that's pretty much it from the Q&A side. You guys can see Dr. Kazi's email address towards on the last slide. So if you have any question, you can reach out directly to him as well. With that being said, I think we would be ending this session. Once again, thank you very much, Dr. Kazi, for your time, for taking our time on a weekend. And I hope this was an insightful session for all the participants as well. And thank you to all of you as well for joining in. And hopefully we'll meet again in the next session with another interesting topic. So thank you all. Have a nice rest of your day.
Thank you so much, Faana. And thank you, Zaid and Ahmed. Bye.
All right. Thank you.